Well, I'd like to thank ARM for this opportunity and hopefully convince you that gamma delta T cells offer a very promising cell type for adoptive cell therapies. So I'd read this to you, but I only have 15 minutes. As a way of introduction, I'm Ken LaMontagne. I run the business development group, and here shows you the experience leadership, quite diverse. Our co-founders, Will Ho is from Wall Street, equity researcher, and Larry Lamb is an academic professor who joined us as CSO and has really led to some pioneer work in the gamma delta field, and one of the first to really grow them ex vivo. So this is our universe of cell therapies as of today. If you squint, you'll find your company. And within a given month, this will change. There'll be less of us, there'll be more of us, or there'll be mergers and acquisitions. So quite dynamic array of companies here. So gamma delta cells offer the best of innate immune responses as well as adaptive immune responses. They sort of sit there in between as a bridge. They have a lot of unique characteristics. They're certainly very plastic. They are a frontline defense, but then they also have the ability to not recognize self, but go after tumors. And they also are wonderful about recruitment of other immune cells to fight off infections as well as tumors. So what about gamma delta T cells? Why are they interesting? Well, the study on the left shows a group from Stanford that looked at 25 malignancies. And what they found here is that the overall prognostic for clinical outcomes is revealed through higher abundance of gamma delta T cells. And certainly it translates into solid tumors as well. So in the middle is Larry's original study that shows that in AML, overall survival correlates well with the amount of gamma delta cells in those patients from high to low. And then lastly, a study done in CTC, colorectal cancer, also shows that high levels of gamma delta leads to a significant difference in overall survival. I want to spend a little bit of time on our platform, which we call Delta X. So we have advanced expertise in ex vivo expanded gamma delta T cells. I'll show you that we can differentiate both VD1s and VD2s. We have a very intellectual property surrounding a first-in-class gamma delta T cell engineering, which we call DRI. This is for drug resistance immunotherapy. What we've done originally in GBM cells is we've overexpressed the MGMT gene of interest. And as you know, with GBM, the standard of care is TMZ. And what MGMT does in these expressed gamma delta cells is conform resistance. So you can't kill them, but you can kill all the other immune cells. So we have a pure population of gamma delta cells attacking the tumor, and I'll show you some data there. And we want to make this applicable to other solid tumors as well, using chemotherapy as a backbone. And then lastly, talk a little bit about our closed system of manufacturing. And also we've, a few months ago, released a press release showing that we have iPSC-derived gamma delta cells as well. So very exciting area for off-the-shelf therapies. Here's our manufacturing schematic for our GBM. The first INB 200, it's called, is a product that's autologous. And it comes from the patient and very similar to all the alpha-beta autologous products. You know, there's a certain process time, vein to vein, followed by freezing, and then followed by administration using a catheter. That's pretty standard for GBM. Excitingly, we've shown that we can differentiate iPSC-derived gamma delta cells to both VD1s and VD2s. VD1s are predominantly in the skin and mucosa, and VD2s are found in the peripheral. So there's differences in homing between these two subtypes. And then upon selection, we could actually do some gene engineering. We have CRISPR-based technology that we can knock in or knock out certain genes of interest accordingly to whatever indication we're interested in pursuing. So a few factors in developing gamma delta T cell therapy. So there's the subunits and then there's iPSCs. So we have all three in store. We have autologous treatment, allogeneic, that's donor-derived, as well as iPSCs. And these gamma delta cells are very promising in adoptive cell therapies for both heme as well as solid tumors. Here's our pipeline. INB-200 is the furthest along. This is in glioblastoma, unfortunately horrible disease. Mean survival is almost about a year. And the approach is now, like I mentioned, the MGM2 gene is overexpressed. So this is engineered gamma delta cells that have that gene overexpressed. That conforms resistance. So the standard of care is TMZ. That wipes out cancer cells as well as normal cells, and then we administer our autologous gamma delta cells. And that's showing really good promise, and I'll show you some clinical data in a minute. Next up is INB-100. This is allogeneic cells, gamma delta cells that are unmodified. So they're just a pure population of gamma delta T cells. And then what we hope is to expand to other programs, our INB-400, which we're hoping to get an IND before year end. We'll feed off of the INB-200, which is autologous, and we're going to have that along with an allogeneic arm. So we'll be the first gamma delta T cell company that will demonstrate autologous versus allogeneic, sort of head-to-head in the same disease indication. Also, we have some other ongoing programs, plus or minus checkpoints. Well, I'll introduce you to the non-signaling CAR, which we're very high on, and we have intellectual property around. And then lastly, our iPSC gamma delta T cells are still early days, but we're trying to scale that up for GMP reasons. So here's INB-100. This is the AML study using unmodified gamma delta T cells. So as you can see, these patients, and there's 3 here, we have now a 4th and enrolling a 5th. They receive a hematopoietic stem cell transplant, and with the same donor that's haplotyped, we recover gamma delta cells from them and use that to engraft. This is a one-time treatment in the circles, and as you can see, followed out, there's no indication of GVHD or any type of grade 3 adverse events, no CRS, and you can see they're out to 2+ years for 2 of them and 1 year for the other. So very promising. These are unmodified gamma delta cells. So we're pretty enthusiastic about this potential outcome. This is the GBM trial, INB-200. We're seeing some long-term durability, followed by surgery and radiation. There's 1 dose in the cohort 1 in blue. So a single dose, and these patients have gone beyond their life expectancy. And dose cohort 2, we're doing repeat dosing, up to 3 treatments, and we've gotten stable diseases. The first one being treated is out a full year from now. So, really promising. Again, no ICANS, no CRS. Gamma delta cells differ from alpha beta cells in that they don't elicit IL-6, which is the problematic cytokine for alpha beta cells, leading to cytokine release syndrome. So we got a lot of benefits using gamma delta. And again, very promising data. We're going to roll this over to our INB-400 and have 25, so a multi-site clinical trial. Yes, gamma delta T cells do infiltrate upon treatment. So on the top left panel is primary GBM, and it's scattered throughout the T cells. But then when you resection the brain tumor, you get a lot of gamma delta T cell activation in that area of tumor biology and tumor tissue. Here's our other program, preclinical program. So this is a non-signaling CAR. So the diagram here shows, as an example, chlorotoxin. This is a very highly toxic neurotoxin, actually used to do tumor painting for surgical resection. So you can see the cell type here has the MGMT gene in it. It's going after GBM, so you could deplete all your immune cells except for gamma delta. We have a non-signaling CAR, meaning that it doesn't have CD3 zeta or the 41BB, CD28. So this acts basically to home into whatever receptor is mediated. So in this case, it's chlorotoxin, which binds to specific chloride channels only within the tumor for GBM and other solid tumors. And this is engaged with the gamma delta receptor, and this leads to cell killing, shown here, GBM plus chlorotoxin, and then normal cells plus chlorotoxin. We've made a CD19 as a proof of concept, so which is interesting, CD19 non-signaling, so it binds to all B cells that express CD19. It won't harm the normal cells, so you don't have B-cell aplasia. That's one of the side effects of the alpha-beta CD19 CAR Ts. But it will home in and attack the tumor quite efficiently. So, and we're excited by that. And there's, gamma delta cells have unique homing properties. So not only can you attack the B cells with CD19, but potentially ones that don't contain CD19 that are in the tumor bed, you can have these bystander effects. And so that's quite interesting. And we're following this up. And if you have a target of interest, we'd love to hear from you and potentially partner. So this is how we perceive gamma delta T cells. We're front and center. We have great characteristics of both the innate and the adaptive immune properties, and really hold a lot of promise because of their plasticity. And then lastly, just want to, why In8Bio? We're a public company. We've been trading for about a year and a half now. We're certainly recognized leaders in gamma delta T cells. We were the first ones into the clinic. We know how to manufacture them. They're not trivial. A lot of groups have struggled with that. We've just announced iPSCs where we can differentiate between VD1s and VD2s. We have 2 clinical candidates, one in AML and one in GBM, and we're hoping to expand that to 4 preclinical programs. So a lot of excitement later this year at ASH. We'll show you more data on the AML, and then breast cancer, later on at SNO, we'll show you data on the GBM. So quite exciting times. I hope I convinced you that gamma delta cells should be recognized as another alternative adoptive cell therapy cell type. So thank you very much.